So it's a bit early for some of us. For Ralph, this is probably mid-morning. Ralph is notorious as an early riser, so it's fitting that he should kick us started because he's going to be the most awake and the most energetic getting started out of the gate. Ralph earned his Ph.D. at Oregon State University, and following postdocs at Emory and at St. Jude's, the latter with Peter Doherty, Nobel laureate, Ralph joined the CDC in 1997, and we were fortunate enough to recruit Ralph to UGA in 2004, where he is a GRA, Georgia Research Alliance Eminent Scholar. Now, I'd like to share with you a mental image I had last night during Dr. Palmer's address, and it was an image of silos, not surprisingly, multiple silos spread out over an open area, and on top of each silo was a physician or a public health scientist or a research scientist, a physician, and they were all on their own silos screaming across to each other unsuccessfully, no communication going on. I don't know if that was because they were all talking at the same time or it was a problem with the terminology or they were just too far away to hear one another. My image had only video, there was no audio. But I think that image is, first of all, it reflects one of the many challenges we have with One Health. But secondly, it's a good segue into our first talk this morning, because with the breadth and depth of his science and the energy and the quality of his work, Ralph is one person who has effectively been able to communicate across many silos, and I'm sure that will be very clear this morning. So without further ado, Ralph's talk is entitled Emerging Viruses from Influenza to Bas-Congo Virus. Ralph, thank you.
Thanks, Duncan.
I've got 2 pointers here, and they're both equally weak. So
I can't see what I'm doing without my glasses. Can you just put it on the main thing?
Yeah, sure.
Thanks, appreciate it.
We've got the blind leading the blind here.
Alrighty, I want to begin by thanking the organizers for inviting me to speak here today. I know it's early for a lot of people. What I'm going to talk about is a story today about emerging infectious diseases, primarily focusing on viruses. Talk about how we've developed some novel platform that's been able to translate across many different venues to where we're at the point now of One Health and looking at biosecurity and food security. And so for those that aren't familiar with emerging viruses, these are essentially viruses that are newly discovered or have the increasing incidence of coming out of hiding, if you will, or they have potential. And you can see over here, this is from the WHO, and this essentially shows that over a period of 1994 to 1999, a variety of infectious and re-emerging diseases occurred, from anthrax to diphtheria, malaria, yellow fever, and so forth. And this happens on every continent all the time. And so this is obviously why we're here today, to talk about this One Health. And what I want to talk about regarding the subtitle of Influenza to Bas-Congo is, why do these viruses emerge? And I have a colleague whose name is Stuart Nickel. He's the chief of the Special Pathogens Branch at the CDC. And I've asked him this question before, as has the press, and essentially he noted that we live in a world that's of rapidly changing global landscapes and environments, and viruses with RNA as their genetic material can adapt and exploit these varying conditions, primarily because of the high error rates in their enzymes and how they replicate. And so he believes that it's no surprise then that the majority of these emerging viruses are of the RNA variety. And so that brings us to the Influenza to Bas-Congo story. And essentially, I'm not going to talk much about this virus, but I'm going to be focusing today on the influenza viruses. And so flu, of course, causes epidemics yearly across the world, has substantial morbidity and mortality. And there's 2 main types of flu that do this, type A and B. And on this virus are a variety of proteins, the hemagglutinin and neuraminidase, that allow for various types of different influenza strains to circulate. The Bas-Congo, for many people who aren't aware of this, is actually a novel rhabdovirus that was discovered in 1999. It has a rabies-like appearance, but it actually behaves like Ebola virus. So it causes a hemorrhagic fever. And this is a nasty little bugger. Fortunately, there really hasn't emerged past 1999. And so it's sitting quiescent, much like the SARS coronavirus came and went. So we've got to be aware of these things as we move forward in the context of thinking about disease intervention. So what's the context of the problem with flu? Well, today I'm going to talk about really our approach to One Health, because flu is a continuing threat to both agriculture and humans. avian influenza virus and high-path avian influenza virus outbreaks occur each year. And essentially, there's greater than 200 million birds affected per year from these viruses. And the consequence is that the cost associated with culling and treating these facilities exceeds the billions of dollars across the world. And now you have swine influenza virus, where there was one serotype of flu for over 30 years, now has a growing problem with all new strains of flu coming in on board. And control of production animals is now exceeding hundreds of millions of dollars per year just in the United States. And what's important is both avian influenza virus and swine influenza virus pose significant health threats because both of these viruses can infect humans. And the novel H5N1 high-path avian influenza virus is now becoming more and more common. There's reinfections occurring every year, and this is recognized now as a significant pandemic threat around the world. So what I want to do today is talk to you about a One Health approach to controlling flu. And what I'm going to talk about is a novel platform-enabling strategy that allows us to actually look at and identify cellular and viral genes that are required for the virus to replicate. And then I want to talk about some novel technologies that we can use for genome editing for the dual purpose of developing flu-resistant animals, particularly chickens and swine. This enhances food security as well as prevents zoonotic spread. And I want to talk about how all this One Health approach actually extends far beyond the United States. It actually is with our collaborators in Australia at the Australian Animal Health Labs. So moving forward on identifying cellular genes, the question a lot of people ask is what is RNA, and I'll just give you a brief overview. This is a natural process that everyone has in their body. It ranges from petunias to humans. It's a process that the host uses to control or silence genes in a variety of different ways. And RNAi works essentially by destroying the molecular messengers that are made that carry the information for proteins. It's important in controlling responses. And essentially how it works is you have a long double-stranded RNA, and an enzyme called dicer binds to this DNA. It cleaves it into a small interfering RNA, and then this associates with a protein called argonate in the RNA-induced silencing complex in cells. This RISC then cleaves one of the strands away from it, and it leaves a guide strand that targets the messenger RNA, and then it cleaves that. So essentially, it's disarming the ability of this protein to make any, this molecule to make any protein. So there's a great need for looking at RNAi as a tool in many different arenas. We looked at it for control of emerging infectious diseases, and we're doing that currently with the Australia group as well, and I'll come to that later. But due to the genetic unpredictability of emerging diseases, having the capacity to make vaccines and respond quickly is almost impossible. So this is nice because we can harness the power of RNAi. It's a synthetic process we can make in the lab. And this allows us to develop new strategies for addressing emerging diseases. And I'm going to give you an example of our success with this. So respiratory syncytial virus is not necessarily an emerging infectious disease, but it is a disease with no vaccine, has very limited therapeutics, and it causes substantial morbidity and mortality in the very young and old. And in fact, by age 2, every child in the world is infected with RSV. 40% of those kids develop manifestations of bronchiolitis and pneumonia and are hospitalized. Those kids that are hospitalized end up getting severe lung scarring and ultimately end up with asthma later in life. So the cost of infection by this innocuous virus is actually huge. So we went after using RNAi to target RSV. Could we silence the virus itself? And in fact, we were able to do so. We are now in phase 3 clinical studies with this virus. So with this RNAi for this virus. So this is the very first RNAi drug that's going on for any virus at all. But therapeutics against pathogens is generally very limited. RNAi therapeutics is limited because the efficacy of delivering these drugs to the right cells at the right time is the black box we have to uncover. And so for some things like macular edema and RSV, as I noted, that's been not a problem. It's been great success stories. And I don't want you to even read this, but you can see from 2004 to 2011, there's been lots of strategies for a whole range of indications and targets, and they're all still in clinical trial. So what does this tell us? It tells us that we need new disease intervention options. We can't just rely solely on old or new biotechnology. We have to think about how we can go after new targets. And so for influenza, one of the major problems is development of drug resistance by the virus. And this has been very clear with the pandemic 0409 virus that occurred in 1999. That influenza strain went from being sensitive to resistance in a period of 6 months. And so this is resistance to Tamiflu or oseltamivir. And what you need to realize is that in the absence of drug resistance, chemoprophylaxis is as good or better than vaccination. And this has been shown in a science study that was published a few years back. So clearly having a toolbox with new and important antiviral drugs is an area we need to look at. So this takes us back to RNAi, because with RNAi we have the capacity now to go after silencing viral genes as well as host genes, the host genes that might be important for virus replication. Influenza is a little small virus that carries 8 segments of viral RNA. And because it's so small, it has to rely on the host and all the proteins that are in the host to co-opt for replication. So when influenza binds to salic acid receptors on a host cell surface, it gets internalized. All the processes from when it encodes and puts its viral RNA into the nucleus for replication relies on all these different host proteins to carry that information forward, to allow for replication, to open up the nuclear pores for this to happen. And then when it exits and it assembles all its proteins at the cell surface for budding, again, a whole nother level of host proteins are required. So we think that host genes that are required for virus replication can be very good targets, and the reason for this is that you can silence these host genes temporarily without deficit to the cell, and moreover, their resistance to mutation. So what I'm going to show you is a little bit of information and a little overview of our SIR RNA approach using the SI genome library. And this is a library that has the sRNAs to silence every single gene in the human genome. So we can therefore ask, what role does this particular gene have in virus replication? And briefly how this works is we essentially have our tissue culture plates that have the sRNAs in there. We overlay our cells on top of these sRNAs, and they're transfected, and 48 hours later those genes are silenced. Then we infect those cells in the absence of the particular genes of interest with a virus and wait 48 more hours so we can measure now the amount of infectious virus it's made, to see if the silencing of the gene allows for more or less virus replication, to look at where in the cell the virus is actually blocked, and so forth. And essentially we can develop then a statistical approach to understand which genes are hits, which ones we should look at that are important for our influenza replication. Of course, this leads to validation, and validation means that we have now developed new sRNAs that target a different seed site on the same gene and say, does this phenotype what we did in our screen? And that is a whole nother process where we do a whole nother screen like this, and then this is repeated twice. So this is a substantial effort, because you're going after every gene in the human genome. You go back and validate it with new sRNAs, and we're looking for the same phenotype. But when we find these, we find amazing things. It's a hugely powerful tool. So I'm going to show you screening results from 2 different libraries, the proteome library, which has 481 genes, and the kinome library, which has 720 genes. And what we did is after screening our genes, we validated, and we found that of the 21 primary hits that we did in our screen, that 5 of the targets, that when you silenced, you got more influenza virus back. So those are antiviral genes, and many of them were previously unknown. They controlled replication inside the cell. 16 of those genes, when you silenced, inhibited the replication. So those were proviral genes. Those were genes that the virus was using to co-opt for replication. And on the same, it's true, at 720 genes, we found 22 hits, 3 that increased replication when silenced, 19 that decreased it when silenced. And the validation step essentially was to repeat this, looking at the same endpoints as virus production, virus replication, virus genome, nuclear localization of the proteins, and so forth. And when you do all this validation, you want to make sure that what you see in one assay complements what you would expect in the other. So if we're seeing silencing of the CPZ gene leads to increased virus replication compared to the controls, as shown here, then you'd expect in the cell the relative levels of influenza genomic message would also be increased. And that's in fact the case. So you're constantly cross-validating. And at the end of the day, this is critical, because if you go this deep into the genome and do all this analysis, you cannot afford to make a mistake early. So at the end of the day, we validated for these 2 libraries 8 genes that were very important for virus replication. Shown in red are the ones that were upregulating flu. So when you silence them, you get more virus back. And for the kinase hits, we found 18 genes that were validated. In this case, 3, that when you silence, increased virus replication. All the other ones were critical genes that were needed by the virus for its replication phase. And I don't expect you to read all this stuff, but all these genes range from cell division to receptors to exosomes. They all have different roles in the pathway that the virus takes in its replication cycle. So what does this tell you? It tells you now we have multiple targets we can go after in the host genome to silence influenza. This whole analysis was also then validated against different strains of influenza, because at the worst case scenario, we can't say, well, this is good for just WSN, which is one strain. We tested all the strains that we had that were important in circulating, and this is the California 0409 that recently has emerged. So what we see here is that the genes that silenced, so we're just showing a handful here, also work for these two different virus strains. So this was important. So now we had new host gene targets for drugs, because we discovered genes that are absolutely required for replication against a whole range of influenza strains. So what can you do with that information? Well, you can drug repurpose, because what you can do now is look for drugs that exist today that are known to intersect with those genes that were identified. And I'm going to give you a couple examples here in a moment, but there's two strategies here. There's taking known compounds and repurposing from new targets, which is what we're primarily trying to do. And then there's known targets that you'd use for new indications, and this would be something like Viagra, which I'll talk about as well. So why do we want to repurpose drugs? What's the advantage? Why not just go through the whole discovery process? And this is the reason. Greater than 90% of all the drugs that go into the funnel fail. And they fail because they have to go through all these steps to get to one drug that goes to market. So you have to put a drug into the funnel, you do your screening, your discovery, then it comes down to your optimization, the chemistry that's going to be required for proper uptake and delivery. The ADMET's really critical, the absorption, the distribution, toxicity. And then everything usually cans in the clinical trials, because you have to have clear indication that the drug has no off-target effects. And finally, you get NDA approval, and you end up with one drug that goes to market. This process is at least 10 years to go from an idea to a discovery to a product. When you do drug repurposing, you already take a known drug, usually it's on the market already. It already has a known safety profile, you know its off-target effects, you know the adverse toxicology that's associated with it. And so I'll give you an example. So Viagra, many are aware of this story, but originally it was developed for high blood pressure in the 1990s. And some brilliant scientists realized that it had a striking side effect, and it helped define essentially a new disease that some men have. And so at the end of the day, Viagra became a billion-dollar blockbuster, and it was never made for what its purpose is used today. But here's another example of taking drug repurposing in our lab. So when we discovered that the O3 gene was important for influenza virus, and this gene, real name is SLC22A, when we saw that this was required for influenza virus replication, we wanted to go after a repurposed drug that can target this. And one of the drugs that was known to be an inhibitor of this gene was probenecid. And probenecid, some people in this room may even use it. It's marketed and FDA approved for the treatment of gout. So a lot of people take this. This is another blockbuster drug taken around the world. So we wanted to see if probenecid could be repurposed. And so typically we did the screen, as I told you, where we reverse transfected our cells and we had all this information. And what we've ended up finding was that O3, when you silence it, reduced virus replication, while its cousin genes, O2, 3, 4, 7, etc., when you inhibit them, actually increased virus replication. And when we silenced O3, we saw, in fact, that we can silence this very well with the siRNAs, and we saw no influenza virus replication. If you silence O1, because it's on the level here where you silence it and it increases replication, we saw more virus than the control. So the early indications were that, in fact, this was going to be a good target to go after. You have to go through the business, though, of proving that probenecid is going to actually work in your model. And so we look to see whether it silenced the message from O3 and the protein levels. You can see that as we increase the concentrations of probenecid in vitro in the cell line model, we saw that there was a decreased relative expression of O3. This showed up here as well, as you can see, and that it had no cytotoxicity as we expected. So everything was good there. And now here's where the rubber meets the road. Does it actually prevent flu replication? And so this is, again, in vitro studies looking at a strain called WSN33 that's at a low versus a high level of infection in this cell line. This is 10 times more virus in these cells. And you can see as we increase the probenecid concentrations, we can see a dose-responsive decrease in the level of virus that occurs in replication in those cells. And these are nice dose-response curves with beautiful ICD-50s. This is the kind of thing the drug manufacturers look for, and we were able to clearly show that this was an effective compound with good IC50 value in this particular model, not only for this virus, but for other viruses that were circulating, like New Caledonia that came out in 1999, and some others as well. So that's all good in vitro, but at the end of the day, does it work in vivo? Will it actually benefit? So we looked at a mouse model where we treated animals 24 hours prior to infection with provinicid, challenged them intranasally, and then looked 72 hours later for the amount of infectious virus in their lungs. And alternatively, we did a therapeutic approach to see if it would work there, where we infected the animals, waited 24 hours, treated, and then looked at the lungs. And you can see here, in fact, it works very well. We see about a twofold reduction in the amount of virus, and that's a huge thing. I mean, because all you need to do to survive flu is lower the threshold of infection to allow your immune system to recover. You don't have to take it to zero. So this was a very good indication that this drug not only inhibited flu for a variety of strains, but could do it in vitro, and it was able to protect from a lethal challenge of influenza virus. So this is a drug that's being moved forward by our intellectual property group, because there's some companies that are interested in that now. I want to talk briefly about other druggable targets, because some of these other druggable targets you can go after with small molecule inhibitors, and we found a variety of different genes that were important, that were required for flu through the RNAi screen that I mentioned. This was CAMK2B, CDC25B, and ICAM1. And for the purposes of today's talk, I'm just going to focus on CDC25B. So this molecule or gene functions in cell cycle by activating cyclin complexes, and really it drives the differentiation of cell cycle from G2 to M, as shown here. And so it also is very important because it shuttles between the nucleus and cytoplasm, as shown here in the replication phase of flu. So as what we're seeing here is that once you have infection, within 30 minutes to 2 hours, all the magic happens where the virus is dumping its genome into the nucleus, which is denoted by this gray line. And then replication occurs, and then all the components then have to get back out of the nucleus. And what they're doing is they're actually going ahead and shuttling on the CDC25B protein. I'm going to show you that data. So CDC25B is proviral. It's one of those genes that when you silence, flu goes away. So that's why it's a good druggable target. And it's showing here that we silence it. We see a loss of replication equal to our positive control, and this is our negative control. What we find here is that there's an inhibitor called NSC-95-397. This is made for a different reason. It was a cancer drug. But again, you're repurposing existing tools now, going after flu. When the target here is CD25B, and treatment results in the inhibition of CDK1 through phosphorylation. And you can see here that as we increase the amount of drug, shown here, we're seeing increased phosphorylation of CDK1 and also ERK, which is a story that I'm not going to get into. And so CDK1, again, is the cell cycle division control protein, and it's affecting this G2 to M transition. So we found that the NSC-95-397 drug blocks this phosphatase activity of this gene. And so we know that there's other genes that this protein works on, including the CDK3 gene. And when we silence that, we use the drug, we see we can silence that gene as well. But here's what's important. This drug inhibitor actually inhibits influenza replication in vitro. You can see that as we increase the level of the drug, we're seeing a dose-responsive decrease in virus copy number. If we look at plaque assay, which is measuring infectious levels of the virus, we again see that as we increase the drug, we're getting a decrease in the level of infectious virus that's produced in the cell line. So the data is very good, saying that this inhibition of CDC25B by this novel small molecule inhibitor can inhibit flu replication in vitro. It has no cytotoxic effects. So to summarize, we saw a reduction in copy number, a reduction in titer, and no cytotoxicity. And then we went into the animal model. I'll talk about that in a moment. But the question is where does this actually block in the cycle? And as predicted, because we know how the influenza replicates, goes in, dumps its magic into the cells, and has to get out, we find that, in fact, between 4 and 6 hours after treatment, we can actually inhibit the virus titers as well. So this inhibitor acts between 4 to 6 hours post-influenza virus infection. When you went and looked at an animal model of infection, again, we're looking at prophylactic treatment where we treat with the drug prior to infection, 24 hours, be infected and look at virus titers, or we treat 24 hours after infection, we see that this inhibitor given prophylactically or therapeutically to the California 0409 strain, this is the pandemic that went around the world in 09, has at least a log, 2 log of decrease as well. So now we have 2 different drugs that I've shown you that can decrease flu. These can be given together, and we can see synergistic and additive effects as well that I'm not going to talk about today. But importantly, this drug, like Verbennis, it also protects from lethal virus challenge. And if we just focus here, this is the PBS control, untreated, and so we see complete protection from lethal challenge. Less so for this particular virus, but still, the mice aren't dying. So now I've told you about an RNAi platform that we use to discover the virus-host interaction, which host genes are important, how we can take advantage of those host genes and repurpose those genes for new targets for drugs. And now I'm going to talk to you about how we can build from this RNA platform and collaborate in a One Health approach. So our One Health approach that we're working on currently is to control influenza virus in poultry and swine to protect food security and, of course, zoonotic influenza emergence. Swine and chickens are the reservoirs for flu. If you can develop resistant animals, you can prevent the circulation and transmission of the virus. We have identified the cellular genes required for flu replication in these animals. We want to now perform genome editing and essentially to introduce traits that lead to commercial products. I'm going to talk more about this in a moment. And essentially, the bottom line is that we'll have a dual impact. This research will increase health and food security by preventing zoonotic spread. It's going to eliminate reservoirs for influenza virus. And this is important because if the pig is considered the mixing vessel for flu. So if we make a flu-resistant pig and it can't get any reassortment going on, you could actually use this as a mechanism to even get around the whole issue of vaccination because the flu won't transmit. We won't get any new reassortments. And reduce production losses, which is an important thing. So our flagship right now I'm going to talk about is with the Australian Animal Health Labs. This is their site in Geelong, Australia, and of course this is where I work at the Animal Health Research Center here in UGA. And when we think about biosecurity and moving from the lab now to the field or to the barnyard, you have to think of biosecurity as risk management. You're managing the risk to the economy, to the environment, to the community. You're regulating pests and diseases and emerging and spreading diseases. So this is what everything happens at CSIRO, the Commonwealth Institute in Australia, and what we're trying to do today. CSIRO is essentially the NIH equivalent. It is the national government for the body of scientific research in Australia. This is all the different sites, and they have sites outside the world, outside of Australia as well. And we're fortunate enough to have a flagship agreement with CSIRO to be one of 3 international flagships around the world. And this occurred recently in 2013. So what are flagships? Well, these research flagships focus on national research priorities. Large-scale multidisciplinary partnerships that are well-funded by world-class experts and having world-class expertise. So they put in $80 million per annum into this whole thing. This is their One Health approach, and they've tied it to UGA, so this provides opportunity to intersect with them now. Why are they even interested in UGA? Well, we have the RNAi platform technology, and we've shared that with them. We have a long history of interdisciplinary collaboration with them, and because we work with emerging infectious diseases, we're utilizing their facilities as well. And of course, the leadership here, David Lee and their group in OVPR was critical in driving this whole process. The portfolio for this biosecurity flagship is shown here. They have various research priorities, animal and vector-borne diseases, diagnostics, and surveillance. In fact, we just had 2 other people come from the Australian Animal Health Labs to visit to develop and use technology we developed with handheld SIRS detection of viruses. They're taking that back to their country to look at Hendra and Nipah virus now. We're working with them on vaccines and therapeutics, of course, resistant animals, which I'm going to talk about, and then, of course, the concept of One Health and medical countermeasures. So I want to summarize all of what this collaboration is about in this one table. So essentially, our biosecurity flagship between the Australian Animal Health Labs, CSIRO, and the University of Georgia has 2 major prongs. One is modified animals for dual benefit, and the other is therapies for One Health. And these are the different tools or subsets that we're using to drive this. So for novel therapeutics, we're combining RNAi and transgenic technology to develop disease-resistant animals, and I'll show you this in a moment. Obviously, influenza-resistant chickens have huge benefit. For One Health, we have this siRNA and RNAi capability to go after emerging infectious diseases, and I've shown you we could do this for RSV, and it provides a rapid response for things that emerge like Hendra and so forth. We have a good immunology program. In fact, we just received what's called the LEAF Award, which allows us to have access to BSL-3, 3-Ag, and Biolevel 4 at their facilities. I cannot get level 4 access anywhere in the United States, even though I was at the CDC. So it's developing these kind of collaborations which drives these kinds of opportunities. So we were looking at innate and adaptive immune enhancers so we can minimize the amount of volume or vaccine that we need. We're developing next generation vaccines for animals because we believe that's an important strategy. Pathogenesis, we're looking at, of course, the ability to utilize their BSL-4 facilities, understand this host pathogen interaction that I've been describing all this morning, identifying critical host targets. This is these genes I've been talking about, these new genes that we can go after. Vaccines, essentially developing tailored immunity for certain animals. Whether you know it or not, bats carry influenza, they carry Hendra virus, Nipah virus, a whole range of different things. Why don't bats die from these diseases? No one can tell me. So this is part of what we have to understand. How can we look at things that we learn from the environment? And of course, looking at different vectors and therapeutics for this approach. And finally, small RNAi biology, this is what is tied to the platform technology I've talked about. Taking the ability to go after and knocking out genes, gene discovery, host pathogen discovery, and actually utilizing this for biomarkers of disease as well. This is where I go every year, or sometimes twice a year, to all. This is in Geelong. And this is their facility. Essentially has level 2, 3, 3-Ag, and 4. They work on a range of animals, cattle, because they look at foot and mouth disease there. They have chickens, of course, pigs, ferrets, mice, bats, you name it. Some of the diseases that are studied underneath this flagship are shown here. For humans, they've been focusing primarily on Ebola and anthrax, because those are major threats. Zoonotics, of course, influenza virus, which is one of our things we work on, but they have a range of other arbovirus, things that are transmitted from insects to man. Cattle, foot and mouth disease. Of course, pigs, we're talking not just classical swine fever, but also influenza and chickens as well. But you can see the impact because the PDRC here on campus works on many of these things as well. So there's good opportunity now to intersect, and of course, they even work on a range of fish diseases. So talking about the small RNAi biology, this is where we can have the capability to input so that we can get some nice output, and this would be looking at new ways for siRNA delivery to target these rapid responses to zoonotic outbreaks, using transgenic resistant animals, developing ways to knock genes in or knock genes out, looking at biomarkers of the disease. And of course, the feedback is all these features that we're working with them today on the transgenic technology, delivery of the siRNAs, microRNAs, this RNAi biology in general. And one of the things I should also mention is University of Georgia is fortunate enough to belong to the RNAi Global initiative. There's 35 institutions around the world that meet together at least once a year, talking about new ways to develop and deliver. So we are in the cutting edge in this area. And of course, having the ability having biosecure facilities. And this is where, now that the ARC is coming fully online, the opportunities arise for this cross-training are absolutely huge, and that's why all has come to us. So RNAi technology for modulation of traits is important because we know in plants disease resistance is very critical. Resistance to rust in wheat, for example, and different cereals that are used. This is ongoing today. We have influenza-resistant chickens. I'm going to show you our proof of principle here in a moment. And the platform capability has many different applications, not just because RNAi occurs in plants and animals, we can go after both major food sources. So this is a dual purpose. We're going to engineer for inherited disease resistance using inheritable siRNA. So essentially, I told you RNA exists in all your bodies. What we want to do is take a chicken's RNAi and refocus it for flu. So we're not adding anything. We're just going to make it target flu. So it's bred from chicken to chicken to chicken. And essentially, when they get infected, RNAi will silence the virus. We also have the capability, of course, to knock out genes that aren't important and restore responses. And essentially, at the end of the day, is developing these resistant animals. So here's genome engineering that was done at all. we've looked at two approaches. I told you about knocking out the genes because we've identified those that are critical for replication. I've just described the RNAi approach where we put these small hairpin RNAis into the genome of the animals that targets chickens. We are now able to actually show we can put GFP in their genome very simply and express them so that their feet and beaks are actually glow. So we know it works. We now have the ability to make a profound international impact, in my view, I think, because we're not changing or modifying anything with the genome of the animal. It's not truly a GMO animal. We have the ability to protect the birds from flu. We can do this today. And we're actually looking at it, and we have the ability, of course, now to reduce the threat of pandemic flu. Because if chickens are the spillover host for influenza, and we know about this, if they become resistant, and we have resistant birds out there, we reduce the ability of the virus to transmit. But you got to look down the road, because we're talking about feeding the future. And whether you know it or not, in the next 40 years, we'll need as much food as we've eaten in the past 500 years. That's a huge effort. This is around the world. And by 2050, we have to double the current food production with the limited resources we have with land, water, and energy. With reduced ecosystem impacts, it's becoming a priority. And then we have to reduce our greenhouse transmissions. That's part of mandate in Australia and other nations as well. And 70% of this extra food is going to have to come from deployment of new biotechnologies because of the threats that exist out there just with flu, wheat rust, and so forth. So feeding the future is an important consideration. So we think that this UGA, CSIRO, One Health theme is a major opportunity and vehicle for engaging, like we're doing today, and actually for global leadership. We're safeguarding right now human and livestock against emerging infectious diseases, looking at flu as one of the primary things, and looking at humans, animals, and the environment. We know that zoonotic viruses are highly unpredictable where they arise and how they spread. We have to have tools to address that. And we know historically, because what's past is precedent, that 75% of the emerging viruses that occur are likely to come out of Southeast Asia. Well, this is Australia. This is right up above them. So having them in place is going to become very important. And these are fruit bats that carry Hendra and Nipah. So the question is, can we see the future where we're going with this? And of course, many people can speculate, but I think at the end of the day, we're going to need the super chicken. We're going to have something that's going to resist flu. And this is probably a year or two down the road, at least in making these in all. So to close, there's been a whole range of collaborators. And of course, our labs, many of them are sitting here today, have contributed at various levels. Mark Tompkins' group, he's showing here with his sexy new glasses. And a variety of different people that have actually come to our lab. Celine has come for looking at the disease enhancement, disease detection. This is John Lowenthal. He drives and organizes the whole thing. He came to UGA, and I was fortunate enough to get him a football game with the president, so this is like his big thing. But I want to thank you for your time, and hope you enjoy the symposium.
Hey, thank you, Ralph. We have time for questions?
Anything? Susan?
I know that a lot of people take medicine. My dad used to take it. He was alive. So, do you have any idea if the use of medicine in the current population and the vaccine for flu will interfere in any way, shape, or form? Are they at risk of not getting good response to vaccinations? Yeah,
that's something that's been brought up actually. So it depends upon your age. The very young and the elderly usually get inactivated flu, so it's not an issue. Live attenuated flu can be inhibited by probenecid. That might in part be the reason why certain vaccine years in certain areas of the country, they don't have good take. These are things that once you make a discovery like this, the CDC epidemiologists go back and do a meta-analysis, and they start looking at these kinds of features. So we don't know the impact yet, but there's clearly lots of things we do every day that impact your ability to resist or flu or other diseases. Yes?
So cancer drugs and gout drugs often have immunosuppressive effects. So does that matter over the course of your
Now, this treatment is, you would give this treatment, you know, at the most for 4 or 5 days. You're trying to stop an acute influenza infection. This isn't like persistent treatment. Gout is a persistent, you treat your whole life essentially. In this case, if you were going to repurpose this drug and someone came in and during flu season with flu symptoms, they tested positive on a rapid detection test, you could prescribe probenecid. And it would have absolutely no major effects because we don't know all about the toxicity of this drug, but it would reduce the amount of flu replication over that period of treatment. So it is effective therapeutically. And perhaps you can give it prophylactically for people that are susceptible to flu, and it may have efficacious activities there. So there's some drug companies, Sanofi is one that's actually looking into some of these things now. Oh, sorry, yes?
You mentioned that the RNAi technology would not be considered GMO. Could you?
Yeah, so what we're doing is we're actually not modifying the genome at all. We're taking RNAi that exists in the chicken today and redirecting its activity. Okay, so we're not changing, we're not adding. We can redirect the RNA. I don't want to get into the technicalities of this. We could talk offline, but we could change its target through a simple modification that doesn't change the RNAi that already is present in the bird. So how it's viewed by the FDA, at least, that's not a serious issue. Taking a gene away is also not a big issue because, as you probably are aware, GMO is a big problem, or could be a big problem in getting a lot of this thing moved forward. People are very sensitive to modifying the genome of food. They forget that Gregor Mendel and his peas have been modifying food since that time and selecting for better quality peas, or cattle have been bred for years to have more or less fat. That's all genetic modification. It's how you view things. It's how you educate people. And you have to go at it like that. If you're resistant saying, well, what we're doing is, you're going to fail. You have to educate. So this is the process that we're doing in Australia now, educating that population. When people see that food is dwindling, a lot of this goes away. So the wheat issue in Africa, Monsanto is selling all their product right now in Africa because there's not enough wheat to feed the people. Here in the United States, they have a very hard time growing this in Nebraska or anywhere else because they're worried about it spilling over to other crops, this resistance. It's people's opinion. I'm of the opinion that GMO has been going on for a very, very long time. It's just the ability to distinguish what is someone that cuts a gene out or versus crossbreeding, you know. It's the same thing. Yes?
I believe that UGA has, like Georgia Tech and Emory, been working on nanotechnology. I wonder if you could add nanotechnology you view in the future predictive One Health of coming along. Because I see a lot of applications that I didn't hear.
Yeah, I have a whole other group in my lab that does nano. And I'm part of the nanosec group here, as well as at Emory and their pediatric nano group. So I do a lot of work in that arena. We look at diagnostics as one thing, because if you're going to say I have a drug to give you, you have to be able to identify the problem, and then you have to be able to show efficacy. So currently today, for viruses, that's why we have almost no drugs for viruses. If you say I have a cold, what is a cold? Is it a coronavirus infection? Is it flu? So today, the ability to diagnose rapidly what someone has and be able to say this person can then use this drug off the shelf and they can give it to you does not exist. So that's where nanotechnology comes in. So one of the tools that we've been working on is a premise of surface-enhanced Raman spectroscopy. Essentially, we can shine a laser light on an analyte. We get back its Raman signature, which is like a molecular fingerprint. And we could tell within 30 seconds what virus it is. And we're working with Duncan on this as well, in mycoplasma and some other areas. That technology, that's why those Australians came here. They're taking that to the field now. We're looking at Hendra Nipah virus. Because part of the whole reason that we can control on emerging viruses is by quarantine and so forth. To give you the example of coronavirus, the SARS coronavirus, I was at the CDC when that happened. We had no vaccines. We didn't even know what the problem was. Where they thought it was human metapneumovirus in many cases. So our lab was busy trying to make monoclonal antibodies and figure things out. But at the end of the day, how that went away was by being able to detect fever as one overt symptom and quarantining people so they weren't affecting others. And then when it silently disappeared. So the ability to detect a problem and know it with high sensitivity and specificity is very important, and that's what nanotech can do. It also is useful for delivering RNAi. We've been able to utilize nanoparticles to have HIV TAT to cross membranes to get in and deliver their payload. The problem is nanoparticle delivery in humans is an issue because most of the nanoparticles end up in your liver. They don't degrade well. So that's another area that has to be overcome. But nanotechnology has huge leaps and bounds in making things move. And again, I could talk to you more specifically offline, but I think for the One Health concept, anything to do with targeting is what nanotech is going to be very good at. And so I think that's happening sooner than later in the technology. Yeah.
I have a question about people being actively treated for gout. Do they have decreased incidence of flu?
Yeah, we actually looked at that. So there is early data that suggests that. Yep. We've actually did a little study at Emory. We're looking with Larry Anderson actually and some others over there now. Flu and RSV, making sure it's specific for flu and not just a general phenomena. All right.
So I have a quick question. So who owns the marketing rights on those green chickens? Because I think we could probably take those to St. Patrick's Day festivals and fund One Health research for quite a long time.
Yeah, yeah. Well, you know, they're actually a good biomarker, those green chickens, because that's another issue that we can make. So I don't know if you guys understand, but flu vaccines today come from eggs. And so one egg equals one vaccine. Well, the issue is we can't make enough eggs, clearly, in the world to provide protection for everybody. So, as again, it's a risk management issue. Who should get vaccines? What level should be done? Should we move to cell-mediated vaccine processes? But we have the capacity to actually, by knocking, as I showed you, knocking certain genes out, we get way more virus back. So from one egg, we can now make 10 vaccines. We've tested that. I didn't talk about that as well. The issue is, do you want to get those chickens in circulation? That's definitely not what you want to do. So having GFP legs is pretty evident when a chicken is one of the ones you're using for vaccine production. So we're actually using that to our advantage now as well. Because those don't go for food, it's just vaccine.
I think you missed an opportunity here with these green chickens. You could have entitled this Green Legs and Ham.
There you go. There you go. All right. All right, thanks, guys. Yep.